Hello everybody and welcome to today's webinar about functional analysis of microRNA. My name is Martin Kreutz and I'm a scientist at QIAGEN's Product Development Department. Before I start with the webinar, I would like to ask you to send me a quick confirmation that you can hear me loudly and clearly. While I'm waiting for that confirmation, I'd like to bring your attention to our legal disclaimer. The QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. For up-to-date licensing information and product-specific disclaimers, see the respective QIAGEN kit handbooks and/or user manuals. These are available at QIAGEN.com or can be requested from our technical service or from your local distributor. Now, so far, okay. Hello everybody and welcome to today's webinar about the functional analysis of microRNAs. There was a small technical issue, so I hope you all can hear me now. And to confirm that, I'd like to ask you to send me a quick confirmation that you can hear me loudly and clearly. While I'm waiting for that confirmation, I'd like to bring your attention to our legal disclaimer. The QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. For up-to-date licensing information and product-specific disclaimers, please see the respective QIAGEN kit handbooks or user manuals. And these are available at our website at QIAGEN.com, or they can be requested from our technical service or from your local distributor. I can see that you can hear me. Thank you for the confirmation. So I'll continue with the presentation. I'd like to start today with a little bit of a background to microRNAs and then go in detail through the different options that are available for functional analysis of microRNAs. Starting with microRNA mimics, which mimics the effect of an endogenously expressed microRNA, microRNA inhibitors, which inhibit the effect of a microRNA, and then talk a little bit about a very special tool, the microRNA target protectors. At the end, I'll say a few things about controls, give a few hints about what you should take into consideration when planning functional experiments, and at the end, I'm happy to answer any questions that you still might have. So, why even assess the function of microRNAs? Why even care? The interest in microRNAs has grown exponentially over the last decade and more, as can be seen in the number of publications about microRNAs, but also simply by the fact of how many microRNAs are listed in miRBase, which is a registration base for microRNAs, which started with only a few entries in its first version, and by now it's way above 30,000 microRNAs that are listed there. With these, of course, are not all human microRNAs, but come from a variety of different species, like human, mouse, rat, horse. Bovine microRNAs are listed, even unusual microRNAs such as from the platypus or microRNAs in plants. Now, what I'm telling you today mostly refers to microRNAs in animals. The mechanisms of microRNAs in plants is slightly different, and doing functional analysis in plants is also a lot more tricky than it would be using animal cells. Now, microRNAs have long evaded the attention of the scientific community because of their small size, but by now we know that changes in microRNA expression can be correlated with gene expression changes, and these often are involved in changes in processes of development and differentiation. They might be involved in signal transduction, infection and inflammatory processes, aging, and in disease. MicroRNAs get a special attention especially from cancer phenotypes, where they are interesting as biomarkers. Most people, when they are looking at microRNA studies, start with doing an expression analysis of microRNAs, seeing which microRNAs are expressed under their experimental conditions, and then see which microRNAs change in expression level when their experimental conditions change. Now, that by itself is already valuable information, but the next step would be to see what does it really mean if this microRNA is upregulated or downregulated. And here's where functional analysis comes into play, especially as microRNAs have a large pool of potential targets. It's not as simple as with an siRNA where you know exactly what sequence this molecule will bind. And with a large number of potential targets due to microRNA's imperfect binding, um, it makes a lot more sense to do functional analysis to see what specific targets are really regulated by that microRNA in the experimental model that I'm interested in. Before I continue, I'd like to remind you of the canonical pathway of microRNA biogenesis. They are first transcribed by RNA polymerase II as long primary transcripts, the so-called primary microRNAs or prime microRNAs. These long transcripts can contain more than one microRNA, especially if they're from intergenic regions. That is interesting to keep in mind because if you know that a microRNA is involved in your process of interest, it might make sense to look at which other microRNAs are transcribed together with this microRNA and might they play a role as well. Now, still in the nucleus, these primary transcripts are then processed by an RNA-3-like enzyme called DROSHA into shorter hairpin-like molecules of roughly 65 to 70 bases length, the so-called precursor microRNAs or pre-microRNAs. These pre-microRNAs are then exported from the nucleus into the cytosol by exportin 5. In the cytosol, they are further processed by another RNA-3-like enzyme, this one called DICER, into the mature double-stranded microRNAs that usually have a length of 21 to 24 bases. These mature microRNAs are then incorporated into the RNA-induced silencing complex or RISC and can start regulating their targets. MicroRNAs with a high homology to the target mRNA might lead to mRNA cleavage, whereas microRNAs with more imperfect base pairing to the target more likely will lead to translational repression and or subsequent mRNA degradation. This is depending on the amount of base pairing, as it seems, but also very much of the composition of the RISC. Now, how does this regulation of genes by microRNAs work? There are several mechanisms suggested. It's possible that there is ribosomal drop-off because the RISC is sitting on that specific site. Then binding of microRNAs might lead to de-adenylation and degradation. And it's also possible that there is microRNA sequestration because microRNAs, once they bound their target, can be transported into the so-called P-bodies and be sequestered there. The incorporation into P-bodies might be a reversible process. Now, how do microRNAs find their targets? What is an actual target for microRNA? It's hard to predict because of the imperfect binding that a microRNA shows. We know about certain mechanisms of binding that are necessary for microRNA target recognition. The most important part of this is the so-called seed region. The seed region covers the first 8 bases from the 5' end of the microRNA. Here, a microRNA has to bind, and from the bases 2 to 7, it has to be 100% match for this microRNA to actually regulate the target. But it's possible that the binding extends further than these 6 core bases, and the larger the coverage is in the seed region, how better the homology is there, the more likely it is that a microRNA will regulate a target. The seed region is interesting because it's the most evolutionary conserved microRNA region. It's the part that's most frequently complementary to the target 3' untranslated regions. and it's often sufficient to confirm mRNA recognition. It's, however, not the only part of the microRNA that will bind to a target, though the mechanisms there and how they affect microRNA regulation are not as well understood as with the seed region. There is for sure binding in the 3' end, and there might also be binding in the central region. This can differ from microRNA to microRNA, but even with the same microRNA from target to target. What happens if a microRNA binds to a target and it is a true binding? The usual process is that this leads to suppression of gene expression, either by translational inhibition or by complete degradation of the target mRNA. In rare cases, however, it has been described that there is an increase of gene expression after microRNA binding. This might be the case in early stages of differentiation. It might also be the case if microRNAs are not binding in the 3' nucleus. Now, if you're concerned how to find potential targets because microRNA binding is so imperfect, there's a variety of different target prediction algorithms available. They are freely available online, and they're all quite good. We listed a few at the bottom of this slide, for example, TargetScan, PICTAR, MicroRNA targets, Diana, miRNA, and Tarbase, which is interesting because it only lists experimentally supported data. How exactly these target prediction algorithms work differs from algorithm to algorithm, but they have a few things in common. All of them take seed region match into consideration. Other points are where is the position of the microRNA binding site in the 3' untranslated region, because the closer it is to the coding region, the more likely it seems to be that a microRNA is binding. Also, it's interesting to look at cross-species conservation, because if a microRNA binding site in an untranslated region is highly conserved, or rather if a sequence is highly conserved in an untranslated region, it's quite likely that it has a function, and that function might very well be that it is a binding site for a microRNA. They also look at central sequence homology, and some of them take also wet-lab research into account, looking at empirical evidence from microarrays or other reporter systems. Now, as useful as these target prediction algorithms are, there are certain pitfalls connected to using them. The first one is that you will receive a large number of potential mRNAs for any given microRNA that you put into the system. And even though this list are long, it might not incorporate all microRNA target possibilities. You will also notice that some potential targets will only be predicted by certain algorithms and others not. So no matter which algorithm you use, you might be missing some of the potential microRNA targets. And the lists do not only differ in content, but also in the ranking, because these algorithms usually give a ranking on how likely it is that a target is regulated by a microRNA, and how a specific gene is ranked often differs greatly between target prediction algorithms. Of course, this as a prediction, it's very educated, but it is an educated guess. So there's also potential for false positives in prediction, and you also should keep in mind it's possible that microRNAs bind certain targets only under certain conditions. Here it's also important to keep in mind microRNAs can be subjected to RNA editing, and this RNA editing, if it occurs, especially if it occurs in the seed region, might alter the potential target that a microRNA can regulate. Now, once you have identified a few targets from the potential list, then you might want to go into functional analysis. Now, one way to go about that is if your experimental model is already well studied, then you might already have an idea which genes might be differentially regulated, or you might know from literature which genes might be involved in the process of interest that you're looking at. And these should be targets that should be on the very top of your list when you're looking at potential targets for your microRNAs and which ones you want to investigate. Now, once you're ready to do the studies, they all basically work the same way. You take a functional molecule that either mimic microRNA function or it inhibits it in one way or another, and these functional molecules are brought into cells. Now, this is in no way a complete summary of all the potential experiments that you could do with functional analysis, but it is a very generic overview of what people often are doing when using functional molecules. The first thing that you can decide is, do you want to directly load a target, or do you want to use a vector-based reporter system? The advantage of a vector-based reporter system is that you usually get much stronger signals because your reporter is always under a strong promoter. Another advantage is that you get a very quick response. Usually you can already see an effect after 24 hours, though you might decide to look at a later time point. And what you would do there in this case is you take your vector-based reporter construct, transfect these plasmids into cells together with the functional molecule, and then perform the reporter assay after the decided time point, which most often is 24 hours. There is some issue with that, however. No matter how your vector-based reporter construct looks like, it is not exactly the same situation as you would have in the cell if you're just looking directly at the target. You might have cloned. Maybe I should quickly explain how these vector-based reporter constructs look like. They usually have a strong reporter, for example, a luciferase gene, and in the 3' UTR of that luciferase gene is a multiple cloning site where you could put in, for example, a microRNA binding site or whole 3' UTRs, depending on how large these are. Now, no matter which way you decided to go, the way how this mRNA, once it's generated, behaves might be quite different to the way how your actual mRNA would behave, because instead of your gene of interest, you're having a luciferase gene there. So as useful and as easy to use these vector-based reporter constructs are, it also you should also consider to at least do to some extent do studies on the actual target, of course. But they're very useful to establish transaction procedures, to get a quick idea, is something happening there, or is nothing happening there. Now, if you decide not to go with the vector-based reporter construct, what you would do is only transfect the microRNA mimic or inhibitor. There's a certain advantage to that because these small molecules are very easy to get into cells. The larger plasmids, for example, for a reporter construct, are much harder to transfect, and they usually require much harsher transfection conditions. If you're using that, optimize your conditions to the reporter construct. It's very easy then to co-transfect the mimic or inhibitor with the established protocol. It's very easy to co-transfect them. That should not be any issue. But you can use much milder transfection conditions, for example, using our hyperfect reagent, if you just want to transfect the microRNA mimic or inhibitor. After transfection, what you can do next depends very much on the microRNA and the target that you're looking at. You might want to do real-time PCR during a pro-RNA isolation. The advantage there is, of course, that you get quick results and that you can look at a much larger number of potential targets. The disadvantage, however, is that a lot of microRNAs do not regulate their targets on transcript level, and that means that you might miss a lot of potential microRNA regulations simply because you're looking at mRNA level and not at protein level. Alternatively, you could look at microarrays or do protein preparation and perform a Western blot to see how the actual protein is behaving. And how long you keep your targets running is depending on really how long you have to wait after transfection is really depending on what target you're looking at, because protein turnover times plays an important role here. We'll take a closer look into that once we're starting to discuss the different functional molecules, and I'd like to start with the microRNA mimics here. MicroRNA mimics are short, double-stranded synthetic RNA molecules that can be transfected into cells and regulate the targets in the same way an endogenously expressed microRNA would. The principle is shown here in a simple graph. We have the potential targets in blue, the mRNAs A, B, and C. And you can see that our microRNA is here in red. The little loop in the middle is just to symbolize that the binding of the microRNA to the target is most likely not 100% perfect. And the binding sites of a microRNA to a target can differ between targets. So the binding site for the microRNA and mRNA A will most likely look quite different from the binding sites in mRNA B and C. And even if you have more than one microRNA binding site on the same transcript, these binding sites might differ greatly. And that can be quite useful, as we see later, to exploit. But for now, it's just important to keep in mind, once a microRNA binds, it will lead to a downregulation of the expression of its target. As I mentioned, microRNA mimics are usually double-stranded RNA oligonucleotides. Here's an example of how microRNA regulation could look like using a luciferase-based reporter system. On the first column, we have a reporter that has a binding site to microRNA miR1, and miR1 is not expressed in the cell line that we're looking at, because miR1 is specific for skeletal muscle. So if there's no mimic present or just a negative control, then you get a strong luciferase signal, here shown in relative light units. If you, however, co-transfect that reporter together with the miR1 mimic, then you can see that the miR1 is binding to the target through its binding site, and you see a strong downregulation of luciferase activity due to translational inhibition via this microRNA. And the third column is, again, a luciferase reporter. Here we used a binding site for Mi16 again, which was endogenously expressed in the cell line on fairly high levels. And you can see a strong downregulation through endogenously expressed microRNAs in this case as well. Now, as I mentioned to you, microRNAs do not necessarily regulate their targets on transcript level, and I'm having an example of this here. We're looking again at miR1, and in this case, we transfected miR1 into these cells, just the miR1 mimic, and later on, after 48 hours, or rather after 24 and 48 hours, we harvested these transfected cells, and we split them. Half of them went into an RNA isolation, and we went to look at different transcripts. And the other half went into a protein preparation, where we later on then looked at the respective protein levels. And here on the right, you can see the results shown in relative expression of this target. Here we're looking at HDAC4, which is a histone deacetylase. And you can see that after 24 and 48 hours, there is no significant difference in the expression or in the presence of these target transcripts. There's no downregulation of the HDAC4 transcript after miR1 transfection. If you, however, look to the left side at the Western blot, then you can see that the protein levels of HDAC4, however, have gone down dramatically in all 4 samples in comparison to negative control. And that is very important to keep in mind. If you're just looking at transcript levels, you might miss a lot of regulation because it often only happens on protein level. Now, if you don't see regulation on protein level after 48 hours, that's not a reason to think that your target is not regulated by this microRNA. It might be just the wrong time to look at. And here's an example of that. Again, we've used miR1 in these 2 cases. And in the upper case, you can see that after 24 hours in comparison to a negative control, the target protein levels have gone down quite significantly. And this reduction in gene expression can be seen after 48 hours, and it stays through to 72 hours, 96 hours, and potentially much longer. Now, if you use the same protein preparation to look at a different target, as we did here, then you can see that for this target, after 24 hours, you cannot yet see any differences in gene expression level. If you look after 48 hours, however, and 72 hours, you can see that there is a clear change in expression level, whereas after 96 hours, this difference is almost gone again already. Changes in gene expression level can usually be measured after 48 to 72 hours, but it depends on a variety of different factors on the microRNA and especially on the target. Here, it depends especially on the protein turnover time. And you might be able to see an effect as early as 24 hours later, but it may take a week or even 2 weeks to see an effect depending on the target. However, for most targets, 48 hours is a good bet, so that is certainly a time point that I would recommend as a starting point if you're looking at changes of gene expression. If you can't see a difference, then you might want to consider doing a time course experiment the way we did here. Now, once you have established that by transfecting a microRNA mimic into a cell, you can see a downregulation of your gene of interest after microRNA transfection. The next step is certainly to confirm, does the same happen if you block the effect of the microRNA if it is expressed endogenously? And to do that, you use a microRNA inhibitor. Now, microRNA inhibitors are chemically modified single-stranded synthetic RNA strands. And here in our scheme of principle, they are shown in yellow. Now, this scheme is a little bit misleading. And the misleading part is that we show the microRNA again in red with this little loop to preserve the make it easier recognizable. But it would suggest that the microRNA inhibitor is not binding with a 100% match, but it is. MicroRNA inhibitors bind the targets that they are designed for with a 100% match. This is important for their function. Now, if a microRNA inhibitor binds to its target microRNA, then this microRNA can no longer bind to the microRNA binding site in all targets that it could potentially regulate. And that means all of these potential microRNA targets can now be expressed. Here's a simple example, again utilizing a luciferase-based reporter system, and we're looking at a reporter that has a miR16 binding site, and you can see that in comparison to a wild type, that's the large column at the beginning, a reporter system that does not have any microRNA binding site, you can see that if a reporter has a microRNA binding site, then endogenously expressed miR16 will lead to a strong decrease of luciferase system. If you, however, put an increasing amount of a miR16 inhibitor, then the inhibitor will bind to the endogenously expressed miR16 and keep it from regulating its target, and the luciferase signal levels go up again. these inhibitors, like the miRNA mimics, are quite stable in the cell lines once you've transfected them. Here we looked at a variety of different time points using different miRNA inhibitors, and they all have been stable for up to 96 hours, which is the latest time point that we have been looking up. They might be stable for much longer. How long they show an effect might depend on the proliferation rate of your cells and the concentration of the inhibitor you used as well. Now, using a miRNA inhibitor can be a little bit more tricky than using a miRNA mimic. Because if you bring a miRNA mimic into the cell, it will regulate its target. And you will see an effect on, at least on protein level, if the miRNA is actually regulating the target. But if you use a miRNA inhibitor, there's another factor you have to take into consideration, and that is that miRNA targets can be and often are regulated by more than one miRNA. If you use a target prediction algorithm to find targets, one thing you can do, for example, is take the name of your gene of interest, put it into the prediction algorithm, and see which miRNAs are potentially regulating the target. And if you had previously done an expression profiling of, for miRNAs, then you can see if any of these potential miRNAs are also expressed in your model system. Because in that case, if these miRNAs truly regulate the target as well, then you might not see an effect even if you bring a miRNA inhibitor into the cell. And we show an example of that here, and in this case we're looking at BCL2, and BCL2 is regulated by both miR15A and miR16. Now miR15 and miR16 are very closely related miRNAs, which means even though they have differences in their sequence, their seed region is exactly the same. And due to the high similarity between the two miRNAs, they can use the same miRNA binding site in BCL2, and they do. So if you use a miRNA inhibitor for miR15, then you will see no change in expression level for the protein simply because miR16 keeps regulating the target. Now, if you use a miR16 inhibitor instead, miR15 will keep regulating the target. Only, and that is shown in the case with the red box around it, only if you co-transfect a miR15 and a miR16 inhibitor, both miRNAs can no longer regulate the target, and target expression levels go up, as shown in this Western blot. So, if you don't see an effect of a miRNA inhibitor, take a closer look at what other miRNAs might be present and see if they also might be regulating the targets in your model system. If they do, all you have to do is use a miRNA inhibitor for these miRNAs as well, and it's very easy to combine 2 or 3 miRNA inhibitors and co-transfect them together and block these miRNA functions to see if the regulation of the target by your miRNA of interest is true. Now, in an ideal case, you can do an experiment like this. And here we're looking at PDCD4, which is the target for miR1. It was one of the targets that we looked at earlier when we looked at the time course experiment. But PDCD4 is not only a target for miR1, it's also a target for miR21. And in the cell line that we've been looking at, miR21 is expressed actually, which means that PDCD4 can still be expressed, but its expression level is lower than it might be if miR21 is not present. And this experiment can show that. If you look at the left side of the Western blot, then you can see that by transfecting a miR21 mimic into the cell, increasing the level of available miR21 in the cell, then you can see that the protein levels go further down in comparison to the all-star mimic, which is our negative control. If you, however, bring a miR21 inhibitor into the system instead, then you can see that protein levels go up because now the miR21 inhibitor is blocking the endogenously expressed miR21, and it can no longer regulate the target, again in comparison to the negative control shown on the right, which is the all-stars control. Now, using a microRNA inhibitor is a very useful tool to decide if a microRNA is truly regulating a target by using the inhibitor on an endogenously expressed microRNA. There is, however, some difficulty with it, and that is a mere inhibitor will completely remove the microRNA from the equation. You will not know which of the potential microRNA targets have been actually responsible for the effect, the final effect that you're seeing. And here you need a more fine-tuned tool, and this is when we're starting to talk about the microRNA target protectors. Target protectors, again, are single-stranded, synthetically modified RNA molecules. And they just do not target microRNAs, and that is the main difference between an inhibitor and a microRNA target protector. A microRNA target protector binds to the microRNA target site, thereby blocking the access of a microRNA to this target site. Now, we've learned earlier already the binding site for a microRNA can differ from target to target and even for different binding sites in the same target, which means if you bring a microRNA target protector into the system, then the target will no longer be, the microRNA will no longer be able to regulate that target, but its regulation of all other targets stays intact. So the microRNA will still regulate those targets, with the only exception that if one of these other targets would have exactly the same microRNA binding site, which is not very likely, then of course the target protector would protect that target as well because it's sequence space. And as you already guessed, here the microRNA target protectors are shown in green, sitting on the target, in this case mRNA A, and here protein A levels would go up because our microRNA of interest can no longer regulate the target, but mRNA B and C would still be regulated, so protein B and C would show no changes in gene expression. Here's our first set of experimental data, again using luciferase reporter responding to endogenously expressed miR16, and you can see that in comparison to an unregulated reporter, expression levels or luciferase levels, activity levels are very low if there are only low amounts or no target protector present in the system, because the endogenously expressed microRNA is regulating them. But with increasing amounts of target protector, these transcripts are protected and luciferase activity levels go up accordingly. Now, if you look at a target of a microRNA, then you have to see how many potential binding sites does microRNA have in a target. If there's more than one binding site, you might need more than one target protector to see an effect, depending on if the binding sites are true binding sites or not. And that is already a finding in its own to see, are all of these binding sites that we're looking at in a target functional. If, however, your target microRNA has only one binding site, as we had in our theoretical models A and C, then of course one target protector is enough. And it's also enough if microRNAs share the same binding site, and that brings us back to the model system that we looked at before, looking at BCL2. You will remember that miR15 and miR16 use the same binding site to regulate BCL2 expression levels. Now, you need two microRNA inhibitors to regulate both, but if you use a target protector, you only need one target protector to protect this binding site from both miR15 and miR16, and that is shown here in the Western blot below, where you can see that if you bring in a sufficient amount of target protector into the model system, then you will see a strong upregulation of BCL2, because this one target protector protects against both binding sites, both microRNAs. Now, you might wonder if our target protector is really specific, and they are highly specific for the binding site that they're used again. And an example of this is shown here. Here we're looking at two different targets, again, of miR1. And what we did here is transfect miR1 into the cell in the presence or absence of a target protector that is designed against the binding site of miR1 in the ADAR gene. Now, if you look to the left side, then you can see that in the presence of the target protector, the regulation of ADAR by miR1 does not happen. The protein levels stay high. If you use the same protein preparation and now look at another target of miR1, which in this case is HDAC4, then you can see that the HDAC4 protein levels go down even if you use increasing amount of the target protector, because the target protector here is designed against the binding site for ADA, a miR1 in ADAR and not in HDAC4. That's why you see a rescue of protein levels of ADAR after miR1 transfection, but not of HDAC4. It does not necessarily always take Western blot or real-time PCR to see microRNA effects. Sometimes you can already notice them with light microscopy. Here, for example, we see an effect of miR9 on certain cell lines. It doesn't happen in all cell lines, but in this particular cell line, we noticed that transfecting miR9 led to an almost complete loss of these cells because it is targeting a cell viability gene. So after transfection of miR9 in comparison to untransfected, you see a large increase of cell death. If we, however, design a target protector against the binding site in that cell viability gene, then you can see that this cell death does not occur because the target protector protects the cells from the effect of miR9 by protecting that cell viability gene. Now, you don't only have to take my word for the effect of these microRNA target protectors. They have already been used by a variety of different groups, and I'd like to show two quick examples here of cases where people used our target protectors in literature. The first one is a study from Nakashima et al. It was published in 2012 in the Journal of Immunology. And Nakashima et al. Have been looking at IL-17 signaling pathway and how impairing it contributes to the increased collagen expression in scleroderma fibroblasts. Now, I'm not going through the whole publication, just looking at the one experiment where they've been using target protectors. And they have seen that IL-17 is regulating the expression of a microRNA, and that one microRNA is miR129 5P. Now, on the left side of this experiment, you can see that after transfection of a miR129 5P mimic in comparison to a negative control, you see a strong downregulation of the collagen gene. If you, however, bring in a target protector, then you can see on the right side that the miR129 5P target protector protects the collagen gene from regulation through miR129 5P. By doing this experiment, the group could show that in previous experiments that miR129 5P is regulated by IL-17, and they can show that by that the regulation of the collagen gene is happening via miR129 5P, and it's happening through the binding site that they designed the target protector against, because they could protect otherwise the gene from it, and thereby showing that the signaling pathway goes via IL-17, miR129, and then looking at collagen levels. Another study, also from 2012, was done by Takeshita et al. It was published in the International Journal of Oncology, and they looked at the effect of miR203 on the migration and invasion of esophageal squamous cell carcinoma, and how it works by regulating LASP1. Now, they already knew that LASP1 was involved, and here it's interesting because they have not been looking at protein levels, but in this case, we are looking at transcript levels, and you can see that on the right side, in the presence of miR203, if you look at the target protector versus the negative control, you see a strong increase of the LASP1 transcript if the target protector is present because it protects the gene transcript for miR203. Now, as I mentioned at the beginning, I'll conclude today's session with a few tips and factors to take into consideration and a few words on controls. Of course, controls are vital to any experiment you're doing, but functional studies rely on a good choice of the different controls that you should have in every functional experiment that you do. The most basic control, of course, is a negative control. And as a microRNA mimic negative control, we could recommend the All-Stars negative control siRNA. Now you might say, wait a minute, this is an siRNA, but not a microRNA. But the main difference between microRNAs and siRNAs is the sequence. They're both double-stranded RNA molecules. So there's not a big chemical difference between them. So it makes siRNA control a viable negative control. The advantage of the All-Stars negative control is that it has no homology to any mammalian gene, and it has been validated for non-effects in both microarray and phenotypical assays, and it has been validated for risk entry, making it a very valuable control. If you're looking at an inhibitor negative control, we recommend the mRNA inhibitor negative control because it targets a sequence of our All-Stars negative control siRNA, showing that it again has no homology to any mammalian gene. Now, if you're looking at positive controls, it gets a little bit more tricky. We usually recognize to use miR1 and a miR1 mimic. miR1 is interesting because there's a variety of different targets that have already been described for miR1, both on protein and on transcript levels. Some of them we've already seen today, for example, HDAC4, PDCD4, or ADAR, which are all regulated by miR1. Another nice part is that miR1 is not expressed under most culture conditions. It's only expressed in skeletal muscle cells. So unless your cell line is derived from a skeletal muscle, you might be safe using miR1 as a positive control. If you, however, use a muscle cell, then there are a few different microRNAs that are still suitable as controls because they have been described as also tissue type specific, so they should not be expressed in your model system, and there have been potential targets for them already described, proven targets in literature. One of them might be miR124, which is expressed in brain cells, or miR122, which is typical for liver. Now, if you're looking at a positive control for an inhibitor, we recommend to combine miR1 inhibitor together with miR1 mimic. The advantage there is that you already have an established system from your mimic experiments, and if you co-transfect a microRNA mimic together with an inhibitor, then you can see if the inhibitor will inhibit the microRNA effects that you should be seeing otherwise. Another important control to use is a transfection control. And in a transfection control, you prove that your transfections were actually successful. People like to use fluorescently labeled microRNA mimics or inhibitors for these experiments, and these are not bad controls. But I don't like them overly much because unless you're doing confocal microscopy, you might have trouble proving that your microRNA actually entered the target instead of just sticking to the membrane. And it requires fluorescent microscopy. And this is the reason why we like our transfection control, which is the cell death control siRNA. And the name already hints at it. What it does is it kills cells, which means if you transfect this control and you see a massive increase of cell death in comparison to your negative control, then you know that your transfection has been successful. The cell death control is a blend of different siRNAs targeting cell viability genes, and it's highly successful. And you can see the effect in light microscopy while you're looking at your cells. There's no extensive fluorescent labeling or fluorescent microscopy necessary. Now you might notice that one popular control is missing in this list, and that is the so-called mock control. A mock control would use a transfection procedure, but without any nucleic acid in the system. Now, for certain transfection procedures, that is a very good control. For example, if you're doing electroporation or a similar approach. But most people use liposuction for transfection experiments, and I don't like the mock control overly much there. The reason for that is that if you look at the way these complexes, these transfection complexes form, they rely on the negative charge of the nucleic acid. Now, if you remove that from the equation, then these complexes might form in a different way. And the mock control I've often seen might behave wonky, whereas all other controls look fine. And you should see the same effect in the negative control, because if you have a negative control that doesn't affect your cell, then it should show the same effect as the negative control. These two should not differ. So if you're using a mock control, you might risk discarding valid data because of a not widely shown chosen control. I'm not a big fan of the mock control, as you might now certainly noticed. Now, other than the right controls, which other aspects might you want to consider? The first thing to consider is, are both the microRNA and the target of interest expressed under your experimental condition? It can be advantageous to have them there, and it can be advantageous to not have them there. If the microRNA is already expressed, it might be more difficult to see an effect bringing a microRNA mimic into the system. You've seen in the case of PDC-E4 and miR21 that it might work, but it might be more difficult to see an effect. So it might be advantageous not to have the microRNA expressed. On the other side, if the target of interest is not expressed, it might be due to microRNA regulation. So not seeing the target might make it easier to see an effect of a microRNA inhibitor that is brought in the system. But having both of them present does not prevent you from doing these experiments. That's why I showed you the experiment with miR21 and PDC-E4, where you can see an increase or further decrease of expression level depending on if you used the right microRNA mimic or microRNA inhibitor. The next question is, what detection system is suitable for me? Can I use a luciferase reporter? And if I don't want to use a reporter, how does the microRNA regulate the target? Is it on mRNA or protein level? If it's only happening on protein level, you cannot use a real-time PCR system to look at microRNA effects. Also, especially if you're looking at microRNA inhibitor experiments, could the target be regulated by other microRNAs? If it is, you might need to either use an inhibitor for that microRNA as well, or you might, if you can, use a different model system where this other microRNA is not expressed. Another part is the chosen cell line suitable for the expected time frame? That is, if you have a strong proliferating cell line, but you want to look at a target that will take a week or two to actually see the downregulation, then that cell line might not be suitable because your cells will be confluent long before you can harvest them. So here it might be necessary, if you have a protein that has a long turnover time, to look at a cell line that's proliferating slowly. And of course, does the microRNA effect affect cell viability? In which case you might need to look into a different cell line or a different model system to see these microRNA effects. So to conclude today's sessions, where can you find all the QIAGEN products that I've discussed today? You can find them all on GeneGlobe, which is www.qiagen.com/GeneGlobe, where you find this field and a search term where you can put in the name of your microRNA, for example, HSA miR16. And once you put that in, you get a variety of different products that we have available for miR16 in this case, which includes the several different PCR assays and arrays, but also what we have available in respect to microRNA mimics and inhibitors. And on our website, you can also find our transfection reagents, which are HyperFect if you just want to transfect a microRNA mimic or inhibitor, or Attractine if you want to transfect it together with a reporter construct. And I'll finally give you a quick overview of all the QIAGEN projects that can help you in your microRNA research, starting with if you want to look at transcript levels, you need to do RNA isolation. If you also want to look at microRNA expression levels at the same time, you can use a microRNA EasyKit, which allows you to look at both microRNAs and depending on what system you later on use, which buffer system, you might also look at microRNA target. And here we are already in the real-time PCR assay field, where we have the MRScript PCR system with the potential to do pre-amplification and specialized PCR rates and primer assays that have been pre-designed for your use, and all of this can be automated. The sample prep in the QIAcube and the real-time PCR assays in the QIAgility. You can look at it in our real-time instruments, the RotaGeneQ, but these MicroRNA assays are also compatible with all real-time PCR instruments. If you have chosen one of our PCR arrays, then data analysis might be made easier using our GeneGlobe Data Analysis Center. And later on, you can do data interpretation using Ingenuity pathway analysis, but also doing functional experiments like the MRScript mimics, inhibitors, or target detectors that I presented to you today. So thank you for attending today's webinar, and I'm happy to answer any questions that you still might have. There's a question on how target protectors don't interfere with the translation. And that is a good question. Target protectors do not interfere with translation because, first, they are chemically modified, so they're not recognized by cellular proteins. All what you have is the sterical blocking of the site to the mimic. But as the target protector is sitting in the 3' UTR, the gene can still be translated because the target protector sits in the 3' UTR anyway. And unlike a microRNA that would have a whole protein complex brought with it that blocks access, in this case, a target protector does not come with any proteins attached to it. So it's just sitting there. And as it's not sitting in the coding region, it does not interfere with the translation. There's a question why microRNA inhibitors and target protectors are used in higher concentrations than microRNA mimics. That is because a microRNA mimic is guided to its target. So once it's incorporated into RISC, the RISC will guide the microRNA to its target. A microRNA inhibitor, however, will have to find its target on its own, so to speak. So you need higher concentrations just to ensure that there is enough inhibitor or target protector present to reach its potential target. And there's another question: what is the best sample to evaluate microRNA profiling like biomarkers in acute coronary syndrome? To be honest, I am not sure. I am not aware of acute coronary syndrome, so that's hard for me to say. And that's more a question about microRNA profiling. Or are you asking about, you already have done the microRNA profiling and you now want to look at what microRNA, what targets might be regulated by your microRNA. If that is the case, I would choose a cell line if you're looking for biomarkers. Are you looking for microRNAs as biomarkers, or are you looking at microRNA targets for biomarkers? Oh, you're looking at microRNAs as biomarkers. Well, that has little to do with functional analysis. What you could do is, of course, if you want to look at microRNAs that are differentially regulated, you need you need samples from where you've seen negative controls, so patients that don't show acute coronary syndrome in comparison to coronary syndrome. But I'm not really qualified to help you with biomarker development. So we have, maybe it's best if you look into our webinar list, because there are specific webinars dedicated to biomarker development for microRNAs from, for example, biofluids or so on. So I would like to refer you to that list. I'm sorry that I can't really help much with you there. And as you're asking this question, one thing I forgot to mention at the beginning of this webinar, unfortunately, is if you're interested in the slides from this webinar, please look into the handout section where they're available as a PDF. Another question is, how can I predict the microRNAs that may regulate my target? Is there an algorithm site for this? There are several sites available. So again, I would like to refer to the slide deck. There is a slide that lists a variety of different algorithms, for example, target scan, miRNA, microcosm targets. All of these algorithms are available to predict microRNA targets. So you would go to the link. There's a link listed on that slide as well. And you can put in your microRNA of interest, and these targets will calculate which potential targets might be regulated for this. So yes, there are target prediction algorithms available, and on that previous slide, we have listed a few of them. Let me see if I can quickly go to that slide for you so you can know what you're looking for. Here, this is the slide that I was referring to, and here you see several different prediction algorithms listed at the bottom, together with the website on where you can find this algorithm. Okay, as there are no further questions, thank you all for attending today's webinar, and I'll hope to see many of you again in other webinars of all the webinars that we have available.